So, we are just going to cover these lymphoid organs, which is a lymphatic tissues which is related to our immunity. So, that is what we are going to start with. It is a general outview of the lymphatic tissues that we are going to cover is that. So, we understand this particular immunity is contributed or covered by this element we call them as lymphoid tissues. Now, what do you mean or what do you understand by the immunity is that the natural sources or added kind of like a strength that helps us to combat the disease or toxins that we are receiving through the environments. Just like when you come out of this room or being in a room, you are kind of like facing challenges against this virus, bacteria, etc., etc. They are present in the air, they are present in the soil, they are present in the material or the substance we are touching in it. And they are kind of like some of the living, some of the cellular morphology is there, some of them are acellular substances. But yet they come into our system, they becomes a trouble for us. So, they are basically a troublemaker. Now, if they are making troubles, thing is one thing, the second thing is that their number. They are plentiful compared to us, we are single man and they are kind of in millions. So, they were we are dealing with millions of our kind of like elements which are going to harm our system. Now, every chance is there to fall in ill because of these elements or these particles, but yet we are not falling it. So, what is basically preventing or helping us not fall in the sickness or diseases because of this particular thing, we call them as immunity. That means that strength that we have inside to deal with these things. So, that particular immunity is contributed by this particular component, we call them as lymphoid element or lymphoidal tissues. So, which are going to give some strength to deal with these invisible enemies that we are dealing with. Now, this particular lymphoid tissues that we have that are going to provide with this particular immunities which are of two types. One we are born with, we call them as innate immunity. Again, after that as we are progressing, as we are still growing, which you guys doing, so these are called acquired immunity. So, you are from the environment, we are still receiving this particular inputs and we are developing our system or combat system. So, this kind of thing we call them as acquired immunity through the environment we are receiving and updating or upgrading the things. They can be of two types, active immunity and the passive one. Active immunity is a sense that means we are building on our own from our system, that whatever the organs and the viscera we have, they are producing the things as there. Passive immunity, we are producing some kind of extra things outside our system and we are introducing that particular element as a booster to the immunity or booster to the system through injections or through capsules or whatever it is, right. So, this kind of immunity we call them as passive immunity. So, these are the two things which are going to contribute to this whole network of immunity systems, right. But yet we are focusing on the lymphoid system. So, we are not talking too much on this particular immunity, rather we swiftly moving to the lymphoid tissues. Now, what these particular lymphoid tissues are doing? They are producing or providing with the business of immunity. So, we understand the functions of this particular element, what they are doing. Now, we have to know the architecture of the system that is present in our body. We have to know what are the different places we are going to find this particular element, which are going to see in subsequent classes or subsequent sessions, right. That is the purpose of this particular system about this particular lymphoid tissue or lymphatic tissues which you are going with it. So, whenever you are dealing with this particular lymphoid tissues, we have to understand what kind of tissue it is. It is a connective kind of tissues that we have to keep in mind. So, we have categorized different types of tissue like epithelial tissue, neural tissue, connective tissue. This belongs to this particular connective tissue or connective tissue network system. That means they are connecting with the systems which has grass and vast ground substances in between. So, their cells are sparse or less and they have plenty of things in between these particular cells, and through the network of the things which are known as reticular things or collagen fibers, they are communicating with this element. That is why you call them as connective tissues, which are constituting of the immune cells, which you call them as lymphocytes, plasma cells, which are network of the things which are present. Now, these particular things are produced, or these particular lymphocytes are produced in the organs, we call them as lymphoid organs. That means they are fractured from or coming from the tissues, we call them as lymphoid organs or lymph producing organs. So, these particular lymph producing organs that we have, they are the production side, which is we are going into it. Now, to categorize these can be classified of two types. One thing we call them as primary lymphoid organs, other is known as secondary lymphoid organs. So, to make it simple, how does it look like? This primary thing, which is where from they are produced, that means say for example is the hospital where these particular things are born. This is one thing. So, just like we are born in hospitals or any maternity setup, we are born. So, that is the point where we have primary begin with. Then we are admitted with the time to the schools, college, etc., etc. So, we get edified or we get this particular educations. So, we get ourselves developed. So, once we are born with this hospital setup, we move to a setup where we develop ourselves spiritually, psychologically, mentally and everything beyond. So, that is the place we call them as this particular schools, colleges, etc., etc., or the sites. So, these things also was born in a place we call them as primary lymphoid organs, which are this maternity place. Then they get matured to reaching to other place, we call them as secondary place. So, those secondary place and the primary place are kind of like different in case of the lymphoid tissues. So, these primary cells are where this born with is of two types or two cells which is predominantly present, we call them as thymus, other is known as bone marrow. Now, out of this thymus which is existing in our body till we reach to the age of 10 years of age. So, this particular site where this particular primary lymphoid organs is produced of, which is known as thymus, is existing only till we reach to the first decade of life. My 10 years of age, this factory is closed, no more productions. So, whatever the things are produced at that time, it is produced over there. Only after that we are left with the bone marrow which are producing these things. Then we are going to get the schooled. So, where they are going to get the schooled after this, they move to the secondary houses. What are the secondary houses? Where they get educated, trained and modified, they are known as the spleen, lymph nodes, etc., etc. So, they move to that place, stay there, grow, take the lesson and be ready, be prepared for helping us to combat with the disease process. Clear? So, this is in short or in a simple words, these things can be classified of. So, primary and secondary lymphoid organs, they are there. Now, out of the secondary which are there, this secondary one maybe have a very localized position. That means they may be very organized or maybe they are scattered. That means they may be just like a clustered in a class or maybe they are scattered all over this campus. So, if they are scattered in all over the campus, they are spreaded, right. So, we call this spreaded amount of this network, we call them as diffuse, that means they are spreaded. And if they are kind of localized in a class, we call them as localized lymphoid organs. So, they are addressing about this particular secondary lymphoid organ, we call them as diffuse lymphoid and dense lymphoid organs. So, we have to know how we are going to find these types of diffuse and where we are going to find this particular localized form of lymphoid organs which are of secondary type. So, this particular diffuse lymphoid organs are of point of interest or point of exam questions that we have to keep in mind. So, where we are going to see the diffuse organs is related to the mucosa. But now, what do you mean by the mucosa? We have this particular mucosa which is covering or layering the inner side of the body, inner side of the tubes which are layering, and this particular mucosa is present in the respiratory system, in the GI system, in the urinary system which are layered by this mucosal element. So, under to that particular mucosal carpeting, we have a collections of this secondary lymphoid organ which are diffuse, that means spreaded. We cannot just like identify, catch this particular part of the gut and see that this is present in here, rather they are spreaded all over. So, that is why we call them as diffuse secondary lymphoid organ and it is related to the mucosa. In short, we call them as MALT, MALT. mucosa associated, and this greatest example where you are going to see this particular MALT is this gut, and in a gut we have to be specific. In a gut we call them as ileum, which is a plentifully, we can see that in a sections about collections of these diffuse lymphoid organs. So, when you see these slides in this region, which you see that these are plentifully seen in that particular MALT. These are MCQs that you have to keep in mind. This is the commonest point where we see this MALT in our body is a ileum. So, this is your MCQ. Whenever you are going to go for a like a MCQ questions, we pick these things up over there, and they are doing the same thing. The prime function is to provide the immunity for the microorganisms or against these particular diseases or the germs that we are going to. So, these are the kind of like a diffuse things, and diffuse one, as you can understand, they are not very localized. That means their barriers are kind of like a also not very defined. So, these lymphoid cells or lymphocytes are there, and they are covered by some kind of capsules. So, we call them as capsules or thin, very non-tangible kind of like capsules which are covering this particular element, which are known as MALT as a diffuse lymphoid organ. This is one thing to cut the long story short. And coming to this particular dense or kind of localized lymphoid, which is having well defined capsule, and they are present in some particular organs like lymph nodes, spleen, thymus and tonsil, which you can catch and see that they are present. Say for example, tonsil is present in the oral cavity or in a place we can see that. Spleen is an organ which is present in this like a part of the abdomen, which is left side of the abdomen. So, this particular thing is present. Lymphoid, we have this lymphoid or lymph nodes are present in throughout of the body, but they have a particular pattern of residence. Like these are the point like inguinal region, we have collections of these things. In case of this particular mediastinum, we have a collections of this. So, we call them mediastinal lymph nodes, inguinal lymph nodes, they are collected localized part, right. So, these are known as thymus. We also mentioned about thymus which is present in the chest wall, chest cavity and etc., etc. So, these are kind of dense lymphoid organ. What is the basic difference between dense and the diffuse? Diffuse is scattered, dense is localized. Diffuse is having a very like a thin or a very poor membrane structure. This is well membrane structure. That means they are localized and well membrane and well architectured structures. These are there. Now, what this particular element has? They have this particular lymphoid tissues. That means the lymph cells are there or lymph producing cells are there. We call them as lymphocytes. These are primary cells. I am not going to talk about cellularity here because that is not point my interest, but just to give an idea about it. So, we have the lymphocytes, and this particular lymphocytes are of three different types. We call them T cells lymphocyte, B cell lymphocyte, and the mature form was the NK cells or natural killer cells. These are modified varieties. They can be classified, these T cells and memory T cells, helper T cells or toxic T cells, suppressor T cells, etc., etc. So, these are the cellular varieties of these particular lymphoid tissues that can be seen, but we are not going to learn about those things in this histo classes. That is why I am skipping this, but we should be knowing that what these particular things are there, T lymphocyte, B lymphocytes. Later on, if that particular things is covered in your curriculums which are there. So, I am just like brushing through. Okay, current gallo? Okay. So, these are things which are going to identify. Now, for us we are going to identify a localized type of secondary lymphoid organs whenever we are going to study in anatomy. So, we are going to study this particular lymph nodes which are present in our body, we are going to study the spleen that is present in our body, we are going to study the thymus and identify the features of them whichever is present in our system, right. So, whenever you are going to identify this particular like lymph nodes or lymphoids element or cells, we are going to identify individual secondary organ and going to identify them as a microstructure. Say if you are going to identify this particular lymph nodes which has got difference with the thymus. So, we have to identify which one is lymph node and which one is thymus, and what basically separates it from identifying from each other. Likewise, the spleen, they have a separate feature or separate or important feature that helps you to identify this one is a splenic tissue which is this particular feature is there, but their prime function as an organ which are of same variety is produce this immune functions, right. So, we are going to see some of this particular lymphoid tissues till this particular projector gets ready. I am just like going through, bring some slides. Coming to this particular lymphoid organs, we are going to see these particular cells which are present in this particular areas, which is we are going to see. This is a point of interest. I am straightly moving into this. One of the example is the lymph nodes. So, which is a collections of this particular like a secondary lymphoid organ, which is a localized variety or dense variety. Lymph nodes, whenever you are going to see this element, we have a particular thing that you have to keep in mind. It is well capsulated, which is present. Under the capsule, we have some kind of space, we call them as sinus. So, subcapsular sinus, which is present in this particular region, and within this particular lymph nodes, there is a flow of lymphatic channels or lymphatic substance that is going in or getting out. So, these are known as the lymphatic channels, which is going in and coming out. Afferent and efferent likewise. Afferent means some kind of lymph nodes moving in, we call them as afferent lymphatic nodules or lymphatic channels, and some of the lymphatics which is moving out, we call them as efferent lymphatic nodules. Now, to talk about this particular thing, histology of this things looks like this. So, this element we are seeing here in a schematic picture is a picture of the lymph node. Now, this is what we are going to see or study whenever you are going to see that in microscopes, they looks like this. So, pretty much difficult to identify this unless until we understand what you are dealing with. So, for that we have to understand this particular element is that capsule that we have to identify, which is present outside, and then we have this collection of this particular dense bodies which is deep to it, we call them as lymphoid nodules, which is the house of the cells that we are talking about. And in between the capsule and this particular lymphoid nodules, we have this space which is existing in here, which are known as sinus, that means the gap. Now, this particular gap is occupied by these particular cells, fatty cells or reticular cells or packing material or collagen things, which are packing these things. Now, apart from that, they are kind of blood vessels may be seen or vessels may be which are getting in and coming out, we call them as lymphatic channels. So, this particular lymphatic channels, through that flow of this lymph which is going through. Now, what is lymph? Which is a ultra extract of this particular blood system or exudate from this, which is extract from this blood system, which is bypassed because of the pressure difference between this particular blood vessels, and it moves to the extracellular substance. So, whatever the plasma it is there, if it is diffusing out through this particular capillaries, it is not getting back 100% through the again capillaries. So, if it is we have this particular arterial end of the capillaries, the blood is moving out, then it is going to the venous end of the capillaries once it going there. So, architecture is that our system of blood flow is that blood is coming to a direction which is coming from the arterial end, and other direction is the venous end. In between we are sitting with the tissues. So, blood has to come to the arterial end and just like shed off everything, then move to that particular space and then climb to the venous system. In between them, they are traveling between the cellular substances. Now, as they are moving through, blood has two elements. One is the plasma element, other is the cellular element. So, this particular cellular and plasma element, they are moving through in between this transition period. Not all of them get through this arterial end of the system to the venous end. Some of them do not find a route, goes through the intercellular substance, and they never find a way to get back to or catch up the train which is going through. So, they deroute themselves through a extra system. That particular system carries this particular plasma or this system. That particular plasma, which is maybe ultrafiltrate of the blood, which is going through and finding a way to reaching back to the blood system through a channels. That what we call them as lymphatic channels. That particular lymphatic channels is a kind of bypass track where this particular leftover of the ultrafiltrate of this particular plasmas, which are going through initially did not get the time to get back to the mainstream, can also get a chance to this reaching to this, which is going in. So, this particular thing helps us in a way. Thing is that this particular bypass creates a thing which attracts this particular like a pathogens to it. So, whatever the things the microorganism entering to our system, they has a tendency to follow this ultrafiltrate, and as this particular ultrafiltrate, they are moving to the extracellular substance and catching up the lymphatic vessels. We have a watchdog element which is present as a lymph nodes. So, this particular lymphatic channels always reach to a nodal point which is known as the lymph nodes. So, as this particular lymph is flowing through the lymph nodes, the bacteria and the pathogens also runs through the lymphatic system, and they get caught at that particular lymphatic channels or lymphatic nodes, and that is the point is the slaughterhouse for the bacteria pathogens. So, this is a kind of like a trap that is being created, or a kind of like a hidden coop that is we are assisted with. So, this is what we are dealing with in our anatomy class, is about this lymphatic system or lymphatic channels, that we understand what exactly it is. It is in short, and you understand already like lymphatic system or lymphatic channels in our 10+2 or before that as well. But for us in anatomy, we are going to identify this particular structure of the lymph nodes which has got these particular patterns. So, whenever you are going to identify them properly, you should be able to identify what exactly they are going to. So, this is how this particular lymphatic system looks like. They have this particular capsule, and this particular capsule, deep to it we have these aggregates of these things, we call them as lymphatic nodules, which are cells, lymphocyte cells. They are different lymphocyte cells, they are present, and then in between them we have some spaces, or in between capsule we have spaces, and here we have these channels. You may be seeing that lymphatic channels, they are opening or going in there, which is constituting the entire structure like this. So, this is how this particular thing should be looking like if you are catching up this particular lymph nodes or lymph node system. So, this is what we are talking about. So, these are the lymphatic channels, they are getting in, then the space in between nodules they are running through, and they get filtered, and they is like screened, and they are going to bypass through this. So, this is about the lymph nodes. Similar to that, another organ we call them as thymus. Thymus is a organ which also have got this particular element, we call them as lymphoid organ, but the difference is thymus is a primary or initial part. We are going to see this particular thymus, they are present in this particular region, and if you remember this particular thymus as an organ, you can always remember this thymus is placed in this particular system. Where is the chart? In this particular lymphoid organ in here. So, this is a primary organ of lymphoid organ. That means, this is the point where it is born actually, right? Where stars are born. This is the point where this particular lymphatic cells are actually born here. So, basically lymphatic tissues or lymphatic cells, they are born in here, and they are, after born, they are sent to the secondary organs which are lymph nodes, thymus, lymph nodes and spleens, etc., etc. So, this is a thymus which is an organ, which is a primary organ, which is a like a birthplace of all the lymphocytes, as you can see, and again it has a capsule, and it has this particular element which is present as a thymic nodules, and this particular thymic nodules has a parenchyma, that means tissues which are present, and they are maybe having separating things which is present inside, and this is how this particular at the spot where this particular organs are like lymphoid cells are produced. Now, this particular thing also have the same thing. You have this particular like capsule, they are having a partition between, we call them a septa, and this particular element can be divided into outer cortex or inner medulla. That means, if you are looking at this particular element close to the capsule, we call them as cortex, midline or deep inside, we call them as medulla. So, whenever you are seeing this, addressing these things, we call them as outer cortex area. That outer cortex means close to the capsule, and if you are penetrating deep, we call them as medulla. So, you should be able to identify and comprehend what do you mean by the cortex part, what do you mean by the medulla part. Cortex means superficial, medulla means deep, and septas, that means partitions. They are made by collagens, fascias, etc., etc. And other point about this particular fascia or this particular partition is that through this partition, blood vessels runs through it, nerves runs through it, all these things runs through this particular system. So, this is about the thymus again. So, we will be identifying this particular organ again and again individually after that. So, cortex, medulla, and how this particular element is running. Then other things is that spleen. So, same thing again. So, these also have got like a same architecture of the things. So, they have this particular spleen tissue, they have a capsule, and they have this particular parenchyma, and this parenchyma can be divided into close to the capsule, deep to the capsule. And the good thing or the differentiating thing about this particular element is that the capsule and this particular parenchyma is kind of like a tight knit structure, and this particular areas have got two basically morphological things. We call them as this pulps. So, they are collections of the things we are seeing there, right. These are the collection things. So, there is a red background there, we call them as red pulp, and within this red pulp we have the non-red areas, we call them as white pulp. So, we have a collections of the red pulp and white pulp. Why this particular red pulp called red pulp, white pulp, we will be coming into it when we are going to identify this particular organ. So, this is the architectural pattern of these things. So, red pulp and white pulp is very much identifying feature for the splenic tissues, and this all this matters to us is that how this particular thing is can be identified of or separated from rest of this particular thing. Other organ, palatine tonsil, that means the tonsil, we have this in our oral cavity. So, this things is present or resided over this particular region. So, we have this particular palate, we have the tongue. So, this particular region we have this tonsil placed in this particular region. So, this particular tonsil also have got same thing which is present. It has a capsule, we have a lining epithelium, we have the space, we call them scripts, and lymphoid tissues. So, each of this particular element has got basic structure or basic architecture like this. So, these are the overviews of the things. Individually, subsequently we are going to identify this. So, this class is about the general idea of the lymphoid tissue, how they are oriented, what do you mean by this immunity which is contributed by this element. We have to identify what do you mean with the primary organ, what do you mean by the secondary lymphoid organ. If you are dealing with the primary organ, what do you mean by the primary organ, what do you mean by the secondary organ, and what exactly they do for us as a primary organ or as a secondary organ. If you are dealing with the secondary organ, what are the sub variety of the secondary organ? That means you may have this particular diffusely present or maybe localized present or densely present. If you have diffusely present, what they are present on, how they are differentiated from the dense variety, that means the localized variety, membrane and dissolvability, and where exactly you are going to see this diffuse variety and where exactly you are going to see this condensed or this particular localized variety, and of that what are the differences that we are going to see whenever you are identifying them individually as an organ. What are the basic architecture like capsule, subcapsular space or these particular nodules or how they are placed, and individually how can it be identified and how can it be separated from each other. Like this things is also lymphoid organ, this is also a lymphoid organ, but how they can be differentiated from one and each other. That is what we are going to learn, that is what we are going to do in subsequent part. Clear? So, this class is primarily theoretical class, I am not just like going to put this individual things right away. Then we are going to move that in the glandular class, we will be going to this particular salivary system next on, but we will be moving back to this particular lymphoid organ individually. So, we are not going to reiterate this particular thing. So, this is the initial introduction which is done, and later on we will be picking up one organ and we will be talking about the general term about that particular organ, we will be covering those things, right. So, that time we will be moving little faster because these are the basic thing. And with the basic we are going to identify the different thing, so like spleen, lymph nodes and thymus or tonsils, that are what we have, right. So, prepare that, see that, study that in your identification point, not much of big things that we have to understand, we have to understand the basic things and how these things individually can be separated up, that is what we are going to learn in the next